Next, we went over genetics. This is Miss Callahan, by the way. And we taught at the beginning of life. Please remember that sperm have 23 chromosomes, egg has 23, and together they make a lovely zygote, which has 46. And when it starts dividing, it makes us. We have 46. Chimpanzees have 48 chromosomes, and that's also known as 23 pairs. In meiosis, you have the same phases that you had in mitosis. You get prophase 1, metaphase 1, anaphase, telophase 1, and then you start into your actual prophase 2, metaphase 2, anaphase 2, and telophase 2. So there they are. At the end product in cytokinesis of meiosis is 4 non-identical daughter cells, so they look nothing alike, and they are haploid, so they have half the number of chromosomes. So for example, an end product of a male that is produced in meiosis, in other words, he's doing meiosis in his testes, he's going to produce a sperm which has got 23 chromosomes. A girl in her ovaries would produce eggs and 23 chromosomes in those. Also remember that meiosis provides crossing over, which gets us genetic diversity. It is known as sexual reproduction because it requires 2 things, or 2 people, to produce this zygote. Intro to genetics. We know genetics is the study of heredity, and that heredity deals with alleles. We get alternate forms of genes from our parents. We get one allele from a mom and one from our dad, and together that gives us a trait. The alleles can be dominant or recessive. If they are dominant, they are going to show up over the recessive, and the only way a recessive allele is going to show up is if it is alone. In other words, you've got 2 recessive alleles from your mom and dad. Gregor Mendel is the father of genetics. He worked with pea plants, if you can remember that. And those pea plants were easy to work with because they had a lot of different traits that he could study. Those alleles, like I said, will code for a trait, and that trait you will show in your phenotype. Phenotype meaning physical appearance. So for example, mine is, I have brown eyes, so that's my physical appearance. But my genotype could be, I might have a blue-eyed allele in there that maybe one of my parents gave me. I don't know. And we did some Punnett squares. So we always represent dominant alleles by a capital letter, and you can use whatever letter you would like. And then a recessive one would be our lowercase letter. So please remember that. Genotype, like I said, would be these. So the big P, little p, that's my genotype. Also, we talked about homozygous. Homo meaning the same. I told you that over and over. Homo means the same. Hetero means different. So if I'm homozygous, that means I have the same. Either 2 big capital letters or 2 lowercase letters. Heterozygous means that I have 1 big and 1 small. So all of these offspring in this Punnett square here are heterozygous for whatever trait that may be. And make sure you know how to do these Punnett squares. So this will go here and over here. And we always do capital letters first just because that's how our language works. So, but this will go down here, across, down, all the way across, down, all the way down, and across, all the way down, and across. Okay. We also talked about disorders. The main disorders you need to remember is cystic fibrosis, which is extramucous, PKU, which is infants cannot digest. We also went over color blindness, which is sex length, and sex length is on your X chromosome. So remember, your sex chromosomes are your X and a Y. XY is a boy, XX is a girl. Boys are more likely to have a sex-linked gene because they only have 1 X, unfortunately. We got 2 girls. So if you got 2, you're going to use the good one. The only way that we get a sex-linked trait is because we got both bad X's, or, you know, both the X's had those alleles on it. Color blindness is recessive, so you have to have 2 bad X's if you're a female to have it, or you have to have just 1 X if you're a male. That's bad. Hemophilia is free bleeder, is what we call it, and it's also recessive. We talked about sickle cell, and sickle is that grass cutting thing that you use. That's why the red blood cells look like a sickle, the top of it like a moon shape. And then we talked about Down syndrome, which is caused by non-disjunction. I've told you that over and over. Non-disjunction, an extra 21 chromosome, and those people have distinct features. Okay. We also talked about codominance, which is pretty much splotchiness. In other words, you're going to see both the traits. If we're talking about a flower, a red and white flower, you're going to see both red and white. Sex length, I just told you about that. It's on the X or Y chromosome. As you can see, our Punnett square's there. And then incomplete dominance, I'm looking for hits. Incomplete dominance would be a blend. So, for example, if you got a red-white flower cross and you get pink flowers, that is incomplete dominance. Pedigrees, here's a picture. If I told you this was a recessive trait, then clearly this female here, which is a circle, is got the recessive trait. So we can pick any random letter we want. Let's say two little b's. She's got two little b's. Here's a male who's also got the recessive trait. He would also have two little b's. These guys would have a big B, little b, or they would have two big B's. In other words, they would have a capital letter in all of their genotypes for the ones that are not colored. Now, if I said this was dominant, then these guys would have the actual whatever it may be, and they would have the big B, little b, or two big B's. Here's a karyotype. This is clearly somebody with Down syndrome because they got an extra number 21 chromosome, but it is a picture. Karyotype is a picture to show us everybody's chromosomes. Not everybody's, but whoever we're looking at the time.